So we start with the question, the stomatal arrangement where there are three unequal subsidiary cells surrounding the God cell. So already we are discussing about the arrangement of the stomatal cells with reference to subsidiary cells in the dicots, in the dicot leaves. So how the in the stomata, in the structure of the stomata, we see two God cells. Okay, usually they are bean shaped or kidney shaped. Those God cells are bean shaped or kidney shaped in case of the dicot leaves. But in case of the monocot leaves, usually they are dumbbell shaped. So now the question is related to dicot leaf, the stomata structure in the dicot. Because in the dicot stomata, there are different kinds of the stomata based on the number of the subsidiary cells and based on the arrangement of the subsidiary cells. See, if the subsidiary cells are more than 3, and if there is no difference between the subsidiary cells and epidermal cells outside in the leaf, that is called as anemocytic. And if there are only 3 subsidiary cells surrounding the God cells, these subsidiary cells are 3 and they are unequal. That means one of the cells is largest, another is the smallest, the second one is medium. So there are 3 unequal subsidiary cells surrounding the God cells. So what is that called as anisocytic stomata? Iso means equal, aniso means unequal. So there are 3 subsidiary cells surrounding the God cells and they are unequal. So the right answer is anisocytic stomata. But there are 2 other options here. In case of the parasitic stomata, the number of subsidiary cells are 2 only. They are parallel to each other. They are parallel to God cells. Those 2 subsidiary cells are parallel to God cells. That is called as parasitic stomata. But if the 2 subsidiary cells are perpendicular to the God cells, then that is called as diacytic stomata. So you should read the types of stomata in the dicot leaves: anemocytic, anisocytic, parasitic, and diacytic. Okay, because these are very important. Next, the question is the experimental organism used by Melvin Calvin to trace C3 cycle is. Melvin Calvin, Benson, Bosham, all these are the scientists. They initially explained the C3 cycle. So it is also named as Calvin cycle. We already discussed that the first product of the C3 cycle, 3-PGA, that means 3-phosphoglyceric acid, that is a 3-carbon compound. So this cycle is called as C3 cycle. And Rubisco is the enzyme. It is involved in the first reaction, that is the carboxylation reaction. It is the most abundant protein. Rubisco is the most abundant protein. It is involved in the first reaction, that is called as carboxylation. RUBP and carbon dioxide. Okay, they are the substrates. And the product is the 3-PGA and enzyme is Rubisco. So here the question is, which experimental organism was used? Here the answers: algae, fungi, bacteria, bryophyta. So you might have known about the chlorella. Chlorella is the unicellular algae. Okay, unicellular non-motile algae. Okay, so chlorella is the answer. They used chlorella because in the experiments, usually simplest organisms as a model, we have to use the simplest organism so that we can understand it clearly. Otherwise, it will be complicated. So they used chlorella for the C3 cycle experiments. And even for the like space experiments also, they used chlorella for the photosynthesis, because in the space they require oxygen. Okay, so algae is the right answer. Next, the classical plant growth hormones capable of cell division are. See, we already discussed auxins, gibberellins, cytokinins, abscisic acid, and ethylene. Ethylene is the simplest gaseous phytohormone. Ethylene in the gaseous form. It is usually used for the ripening. Ethylene, you know. So here, what they have given: auxins, gibberellins, cytokinins, abscisic acid. So out of the four, abscisic acid is a growth inhibitor. Okay, there is no cell division. Abscisic acid, actually, it just used as a switch off, anti-growth hormone. So we can remove that option. And auxins we know, apical growth, apical dominance. Auxins means we have to remember apical dominance. That means due to the presence of the auxins, always growth occurs through the apical bud. The plants grow continuously. Okay, and also phototropism, geotropism. All these we have to remember, they are linked with the auxins. And gibberellins. So these also useful for the elongation, for the fruits, to get the more fruits. Okay, and the cytokinins, they are mainly involved in the cell division. Okay, so the right answer, the classical plant growth hormone capable of cell division. Which are the hormones useful for the cell division? Cytokinins is the right answer. And the next question, the first formed product in Krebs cycle is. So what is the first product? We already discussed about the first product in the C3 cycle, that is a 3-PGA. And the first product in C4 cycle, that is the OAA, oxaloacetic acid. It is a C4 compound. So it was named as C4 cycle. And now the question about the Krebs cycle. Why it is called Krebs cycle? Because it was first described by Sir Hans Krebs. So on his name, it is named as Krebs cycle. So what is the first product? Pyruvic acid? No. Pyruvic acid is the end product of the glycolysis. You know, in the glycolysis, what is the end product? Pyruvic acid, or it is also called as pyruvate. So it is not the right answer. Next, acetic acid, no, it is not at all related. Ascorbic acid, it is not also related. And citric acid, yeah. The citric acid is the right answer. Citric acid is the first product formed in the Krebs cycle. So you also should remember that Krebs cycle is also named as the citric acid cycle. And it is also named as the tricarboxylic acid cycle, TCA cycle. Krebs cycle or citric acid cycle or TCA cycle or tricarboxylic acid cycle. So these are the names given for the same cycle. Why it is called citric acid? Because the first product is citric acid. And why it is called as tricarboxylic acid cycle? Because citric acid is a tricarboxylic acid. It has 3 carboxylic groups functions. There are 3 carboxylic compounds. So it is called as tricarboxylic acid, citric acid. So you have to remember citric acid cycle, Krebs cycle, tricarboxylic acid or TCA cycle. So these are the questions from the 2025 paper for MSc Entrance Botany. So we discussed all this. Now we'll start another question paper, that is 2024 MSc Botany entrance question paper in 2024. This is the question paper. In July it was conducted. It was an online test. So now the first question we see here: asexual reproduction in Oedogonium. Asexual reproduction in Oedogonium takes place by, that is the question. You know, in the Oedogonium belongs to Chlorophyceae. It is an algae. Okay, Oedogonium belongs to algae. So it belongs to Chlorophyceae. And there are two types of the reproductions: asexual reproduction and sexual reproduction. So how the asexual reproduction takes place? Here you see once, in the asexual reproduction from the filament. So it is an unbranched filament. Oedogonium is an unbranched filament. And here one of the cells, they form the zoospore. That structure, the cell is called as sporangium. It forms zoospores. So that cell is called as zoosporangium. What is zoosporangium? If the spore contains the flagella, that is called as zoospore. Okay, since zoospores are formed in that cell, that is called as zoosporangium. Since the spore is motile, it is called as zoospore. So here there are flagella arranged in a circular form. Okay, so here how the question is regarding the asexual reproduction in Oedogonium takes place by gonidia. No, exospores. See, gonidia are related to sexual reproduction. Exospores are not related to this. Since zoospores, usually there is a small mistake here. They should be zoospores. That is the right answer. Autospores, it is the wrong answer. So the nearest, most suitable answer is zoospores. Okay. So here, if you see, these zoospores are involved in the asexual reproduction. These zoospores which are released from the zoosporangium, later under the favorable condition, they germinate and they form the new filament. And now the second question: cenobium is a. What is a cenobium? So it is related to wall wax. Once you see, this is the wall wax. Is there a difference between the cenobium or colony? See, you see the different structural forms of algae. The thallus organization, it is also called as thallus organization in algae. The thallus, the body, the algal body is called as thallus. Usually it is not differentiated into roots, stem or leaves. So that structure is called as thallus. So here thallus organization is different forms. As we discussed earlier, it might be unicellular like chlorella, non-motile. Okay, and multicellular. But here you see, this is a colony. There are several unicellular organisms here, just like chlorella. There are so many unicellular organisms. They form a colony. They are living together. They are independent, just like our apartment. Just like our apartment, high-rise buildings are big apartments. You see, there are several independent houses in the same apartment. They live independently. but they have common like some of the uses. They have some common needs, just like water supply, drainage, like that. All of them use commonly. Maybe a shopping complex. All of them use commonly, but they live independently. Just like here also, there are several independent unicellular organisms, but they are all connected by cytoplasmic connections. They move in a single direction with the help of the flagella. And there is a difference between the colony and synobium. In the colony, there is no fixed number of the organisms. They grow continuously. The colony size may grow continuously. But what is the synobium? In the synobium, there is a fixed number of the organisms. Definite number, it is also called as definite number of the organisms. The number is fixed. They don't grow further. Once a colony is formed, the number is definite. That means fixed. And in volvox, what we see in the synobium, here they have flagella. So it is a motile synobium. So now we come to the question here. Synobium is a non-motile colony with constant number of cells arranged in a regular manner. So here, constant number of cells arranged in a regular manner, it is correct, but non-motile colony, it is wrong, because volvox is an example for the synobium. They have flagella. So it is a wrong answer. Motile colony, yeah, it is right, but with variable number of cells arranged irregularly, that is wrong. The number is fixed, definite number, and they are regularly arranged. So motile colony is correct, but this remaining answer is wrong. So second is wrong. Third one, colony with unlimited cells arranged in regular manner. It is also wrong. Unlimited is wrong. They are limited in number. And the fourth option is colony with limited cells arranged regularly. Yeah, that is the right answer, because the number of cells is whether they have flagella or not. It is a different thing. Synobium may be motile or non-motile. If they have flagella, they are motile. If they don't have the flagella, they are non-motile. But the main feature is they are limited in number and arranged regularly. So that is what we see here. You see, this is the synobium of volvox. Here in the asexual reproduction, inside the volvox, there are so many daughter synobia produced here. That is the answer. And next, isomorphic diplohaplontic life cycle is the characteristic feature of. That is the question. Isomorphic in the algae, because usually in the beginning, all the questions are related to algae. Next, fungi. Like that, they go in that order. So now our questions are related to algae. In the algae, usually you see there are different kinds of the life cycles we observe. So, for example, volvox. In the volvox, what we see, except the zygote, all the stages in the life cycle of the volvox are haploid. So that life cycle is called as haplontic life cycle. And in the Oedogonium, it also belongs to chlorophyceae. Both volvox and Oedogonium belong to chlorophyceae. In the Oedogonium also we see all the stages except the zygote are haploid. So the zygote cycle, the zygote stage is very limited, only for short period. So this life cycle is called as haplontic life cycle. And in Chara, it also belongs to chlorophyceae. In this life cycle also called as haplontic life. But you see, if you come to the ectocarpus belongs to Phaeophyceae, that is also called as brown algae. Here we see there are two different stages. One is haploid stage, that is called as gametophyte. Another is diploid stage, that is called as sporophytic stage. So we find both the sporophyte and gametophyte, both the stages in the life cycle of the ectocarpus, but they appear similarly. Okay, morphologically, there is no difference between the from outside. When we see from outside, they are structurally similar, but in the chromosome state, with reference to chromosome state, one is diploid, that means sporophyte, another is haploid, that is gametophyte. So it is called as, the life cycle of the ectocarpus is called as isomorphic. Iso means same. Morphic, morphologically they look similar. Isomorphic diplohaplontic life cycle. And in Polysiphonia, it is a red algae, also called as rhodophyce. It belongs to rhodophyce. So the life cycle is called as diplobiontic, because it is also called as triphasic diplobiontic, or in some books they write it as haplodiplobiontic, because it has a haploid stage, that is gametophyte. In the life cycle of the Polysiphonia, we find haploid gametophytic stage, and there are two diploid stages, carposporophyte and tetrasporophyte. These two are the diploid stages in the life cycle of the Polysiphonia. You should remember all these terms. Gametophyte is always haploid. Sporophyte is always diploid. In the life cycle of the Polysiphonia, we find two different sporophytic stages. That means diploid stage. Carposporophyte and tetrasporophyte. Carposporophyte is also called as cystocarp. You should remember that term also. Cystocarp or carposporophyte. Both are same. So you have to remember the names of the life cycle. Actually, it was asked in my one of the main interviews, this question. So isomorphic diplohaplontic life cycle is the characteristic feature of ectocarpus. That is the right answer. In Polysiphonia, as we discussed, it is different, and Oedogonium also different. Okay, Cara. It is these two are haplontic life cycle. This is diplobiontic life cycle. Okay, Polysiphonia, triphasic diplobiontic life cycle, and isomorphic diplohaplontic life cycle ectocarpus. It is very important question. Next, nanandria, or what are the nanandria? Where we find this nanandria? In the life cycle of the Oedogonium. In the life cycle of the Oedogonium, there are two different types of the species: Macrandra species and Nanandra species. In the Nanandra species, we find a male filament which is very short in size, that is called as nanandrium. The plural of nanandrium is called nanandria. So what are the nanandria? Long male filaments, it is wrong. Long female filaments, it is wrong. Dwarf, that means short male filaments, it is the right answer. They are called as nano. Nano means small. Nanandria, that means small or short dwarf male filaments. And the other option is dwarf female. So it is not related to female. So right answer is dwarf male filament. Now once you see here. So this is the Oedogonium here. In the Nanandrous species, we find this, and the Oogonium, this dwarf male filament, it germinates. Okay, androspores are produced here. One of the androspores here, it germinates, it falls on the filament near the Oogonium, and it germinates and it forms a male filament. Okay, on the top of this we find the antheridium. Okay, so this is related to Nanandra species. That male filament is called as nanandrium. Okay, next question is chlorophyll E is present in. Yeah, chlorophyll, you know, that is very important question. and there are several questions regarding the chlorophyll. Particularly in the chlorophyll, we know chlorophyll A, chlorophyll B, chlorophyll C, chlorophyll D, chlorophyll E. These are all the different forms of the chlorophyll. There are little structural changes. C55, H72, O5, N4, Mg. So that is the formula, molecular formula for chlorophyll A. C55 is constant in the chlorophyll. See, number of carbons, C55. H72, it is 72 in chlorophyll A and 70 in chlorophyll B. O5, in chlorophyll A, there are O5, oxygens are 5, but in case of the chlorophyll B, oxygens are 6. N4, it is constant. Okay, and you should remember pyrrole rings. There are four pyrrole rings are present in the chlorophyll structure. They are connected by a single metal ion, that is magnesium, Mg+2. So what is the metal ion present in the chlorophyll? It is also most important question. What is the, just like what is the metal ion present in the RBC? Iron. It is not related to botany. But what is the metal ion present in the chlorophyll? That is magnesium. And what is the difference between the chlorophyll A and chlorophyll B? Chlorophyll A has methyl functional group. Instead of methyl, in the chlorophyll B, there is a CHO, aldehyde group. So like that, you have to remember all these chlorophyll structures. And we know that chlorophyll A is common in all the photosynthetic organisms, just except bacteria. Bacteria has bacteria chlorophyll, but in all other organisms we find, for example, in all the algae, whether they are green algae, brown algae, red algae, any type of the algae, they have chlorophyll A. It is common. In all the angiosperms plants also, chlorophyll A is present and chlorophyll B also present. And in the algae, you see, I will tell you, in the algae, there are different groups: chlorophyceae, pheophyceae, rhodophyceae, xanthophyceae. These are the main classes of algae. Here, chlorophyll B is only present in chlorophyceae, that is a green algae. Along with the chlorophyll A, chlorophyll B is present in chlorophyceae, that is green algae. And in pheophyceae, that is a brown algae, chlorophyll A and chlorophyll C both are present. And in rhodophyceae, that is a red algae, chlorophyll A and chlorophyll D is present. Chlorophyll A is common. And in xanthophyceae, okay, that is the yellow colored algae. There we find chlorophyll E. That is the right answer. So the answer is chlorophyll E is present in chlorophyceae wrong, rhodophyceae wrong, pheophyceae wrong, xanthophyceae is right. Okay. So chlorophyll C is present in pheophyceae, chlorophyll D is present in rhodophyceae. Okay, chlorophyll E is present in xanthophyceae. In chlorophyceae, what we find? Chlorophyll A and chlorophyll. Okay, right. Next question is, Floridian starch is the reserve food in. Where we find Floridian starch? Actually, the same question was asked in a 2025 question paper also. Floridian starch. What is the reserve food form? In which form it is stored? Floridian starch in the Polysiphonia, in the Rhodophyceae. Sometimes in the earlier paper they asked in Rhodophyceae. They changed the option. In Rhodophyceae there are different organisms. Bactrachospermum is also present. Polysiphonia is also present. Sometimes they give the option as Rhodophyceae. Sometimes they give the option as Polysiphonia, because Polysiphonia belongs to Rhodophyceae. So the right answer is Polysiphonia. Ectocarpus. What is ectocarpus? It is a brown algae, also called as pheophyceae. So what is the reserved food material? Laminarin starch, the form of the mannitol and laminarin. These are the two reserved food material present in ectocarpus, that is the pheophyceae or brown algae. And Oedogonium, Oedogonium is a green algae, chlorophyce. You know what is the food form? It is stored in the form of the starch, just starch. Char also belongs to chlorophyce. So the reserved food material is in the form of starch. So these are the things you should remember. Next, nuclear endosperm is present in. So now the question is related to embryology. So in the embryology, you see there are different types of the endosperms are present. You see, nuclear endosperm, cellular endosperm, and helobial endosperm. Another type also, ruminate endosperm. That question already we discussed, where we find ruminate endosperm in the Anonaceae or Anona or Aracacataceae, Aracaceae family, and Myristicaceae family. In these three families, we find the ruminate endosperm. Myristica fragrans belongs to family Myristicaceae. Aracacataceae belongs to the family Aracaceae. Anona squamosa belongs to the family Anonaceae. In these three families, we find this ruminate type of the endosperm. Okay. Okay. And now the question is nuclear endosperm. You should know what is the differences between nuclear, cellular, and helobial. Helobial endosperm is the intermediate form between the nuclear endosperm and cellular endosperm. In the nuclear endosperm, what happens? Initially, in the primary endosperm nucleus, that is also called as PEN, there is a division. It forms a nucleus, and there is no cell division or cytoplasmic division. Only nuclear division takes place. This is called as free nuclear divisions, and there are free nuclei. It is called as free nuclei. There are no cell wall formation here. So usually we find this condition in case of the coconut. What is coconut water? That is nothing but the nuclear endosperm. Later, finally, after time at the mature stage, there will be cell division or cytoplasmic division. They form the cell wall. That is called copra. What we see in the coconut, in the tender coconut, we see the water. That is nothing but nuclear endosperm. Okay. And in case of the cellular endosperm, from the beginning there is a cellular division. Total nuclear division is followed by the cytoplasmic division and cell wall formation. So we see here cells, formation of the cells. So it is called as cellular endosperm. And helobial endosperm, here it is the intermediate form. There is in one side there is a free nuclear division, another side there is cell wall formation with the cells. So it is the intermediate form. It is called as helobial endosperm. In the monocots, in the helobiae, we see this condition. Okay, ruminate, we already discussed. So the right answer, what is the right answer? Nuclear endosperm is present in. Where we find nuclear endosperm? Not Anona, not Magnolia. You see, already we discussed in Anona, what we see, the ruminate endosperm. Okay, Magnolia, it is wrong. Okay, and Cocos, Monocoria. Out of this, the right answer is Cocos. Okay, so here you see nuclear. I have given the examples also. You should remember, because now the question is, it was asked like that, but sometimes they change the question form. They may ask a different question from the nuclear endosperm. You should remember the examples for the nuclear endosperm. That is the Cocos nucifera, coconut, Rotalaria, Grevillea, Robusta. So, and also one more question may come. Vermiform Hastoria is found in which plant? In which endosperm? Vermiform. In Grevillea robusta, we find in the nuclear endosperm, it forms a nuclear endosperm where we find the vermiform shape. Vermiform, that form is called as vermiform. Hastoria, a worm-like structure. Hastoria, this new endosperm it draws the food material from the nucellus with the help of the hastoриum. That hastoриum is it is a worm shape, so it is called as vermiform hastoria. It is found in Grevillea robusta. And in Macadamia, it is a papillated hastoria. And cellular endosperm examples, Hydrocera, it has branched hastoria. And in Magnolia, the hastoria is it has two cells only. And in Nemophila, hastoria from both micropylar and chelazal side. It forms hastoria. Usually hastoria is formed at one side, either at the micropylar side or chelazal side. But in Nemophila, hastoria is formed at both the sides. And in Loranthaceae family, composite endosperms are formed. So all these are the examples for nuclear endosperm and cellular endosperm. Okay. And helobial endosperm is found in Trillium and Monocoria. So here the question here is Monocoria. It is example for helobial. Magnolia is example for the cellular endosperm. Annona is example for the this ruminated.